Hi everyone, this lab is about the microscope, so it's going to teach the basic components about the microscope, and it's very similar to what I covered in the microscope and staining overview lesson, so a lot of it is repetitive if you did see that. But it's a good review. So to give background on the microscope, again, we talked about this in the other lecture. Microscopes let us magnify very small organisms, microorganisms, and there's different types of microscopes. We are focusing on the light microscope in lab, and you use this to view microbes that are in the micrometer size. Remember that bacteria are typically measured in micrometers, so this is where we look at E. coli, bacteria, salmonella, many different types of bacteria. Viruses are much smaller, they're nanometers. You cannot use a light microscope to look at viruses. I wish we could, but we can't. We use electron microscopes to see viruses. And the parts of the microscope, I want to go through them and just make sure everyone knows what they're used for, especially since you guys are not actually using a microscope with this online course. So there's the base. The base is where you hold the microscope from. So there actually should off. So here's the base, and this thing is called the arm. So the base and the arm is how you hold the microscope. That's the purpose of them. And the light source is here. It shoots light up so that you are able to eventually see your image because your eyes are here. Here is the power switch. This is turning it on and off. Right here under the power switch is the rheostat knob, and we always want to make sure that that's a baseline of 4.5. You set that up. Okay, if you were actually in lab, a lot of times students can't see anything because that knob is very low, so it controls the amount of light. Here is the condenser. So this condenser right here controls the amount of light that's coming from this area right here. On the condenser, there's a little lever called the iris diaphragm lever that you can move left and right, and it basically controls the amount of light that is shooting to your specimen or your slide. Here is the stage. The stage is where you put your slide on. Here is the stage adjustment knob. So this knob helps move the stage left and right, just goes this way, not up and down, just across. Here's the arm. Again, you hold the microscope from the arm and the base. And then the objective lenses, your microscope, your light microscope has 4 objective lenses. They're 4x, 10x, 40x, and 100x magnification. And then here's the ocular lens. This is where you put your eye, and it is 10x magnification. And here's the coarse focus knob. The coarse focus knob, they're on the sides of the microscope. They help the stage go up and down. The fine focus knob also makes the stage go up and down, but at a very fine level. So when your objective lens is so close to your slide and you don't want to smash it, when you're using high objective lenses such as 40x and 100x, you're only going to be focusing using the fine focus. So here's everything about what we just went over, and the main things I want you guys to know is the condenser right here is what focuses light on your specimen or slide, and then on the condenser is the iris diaphragm lever, and this adjusts the light from the condenser. So the light is here, it shoots into the condenser focuses the light into the specimen, and you can control the amount of light or the level of light using the iris diaphragm lever. Magnification, the ocular lens always has a magnification of 10x. The objective lens, there's 4 magnifications. There's 4x, 10x, 40x, and 100x. Whenever you're using 100x magnification, you'll always add oil. So that's why we say this is the oil immersion. You will never use this objective lens without oil because, okay, we tell students that you'll smash the slide, but the other main reason is that you get better resolution. You actually can see what you're looking at using oil. Magnification versus total magnification. If someone asks you what's the magnification, they're just asking you which objective lens you're using. So the word magnification means which objective lens you're using. The word total magnification means the objective lens times 10 times the ocular lens, because that is the total magnification. So if you had the 10x objective lens and I walked to you in lab and I said, what magnification are you looking at? You would say 10x. If I asked you what's the total magnification, you would say 100x because it would be 10 times 10x. So if you have the 40x objective lens, your magnification is 40. Your total magnification is 400. At oil, your magnification is 100x. Your total magnification is 1000x. And bacteria should always be looked at using 1000x total magnification. So again, these are your ocular lenses and these are your objective lenses. Another review on resolution from the last lecture. So resolution is just the minimum distance that two objects can be separated. And the smaller this minimum distance, the higher the resolution. It's limited by wavelength. Oil immersion, which we use at 100x magnification or 1000x total magnification, is important for increasing resolution. So if we look at an image, and remember, you only use oil at 1000x total magnification. With no oil, you get a fuzzy image because the light is scattering everywhere. And then with oil immersion, you get a very clear image because oil, what it does is your objective lens, your 100x objective lens is so close to your slide. When you add oil between them, you trap all the light so that no light is being scattered. So oil prevents refraction and loss of light rays. Using the microscope, I'm fully aware that you guys are not using a microscope because it is an online course, but just as a review, because I think this will be important if you ever go on to use it, whenever you're looking at a slide, you always start with the lowest magnification in place. So for the objective lens, that's 4x. Then you focus it, and then you go to the 10x objective lens. You focus the slide using the coarse focus knob that brings the slide up. Then you go to the next highest objective lens, which is 40x. And then finally, when you're looking at bacteria, you go to the next highest objective lens, which is 100x. And with this one, you use oil, and then you use the fine focus knob to focus your slide. So the objectives from this lab is I want everyone to know what's the type of microscope we used in lab, light microscope. What units does it measure? Remember, is it micrometers or nanometers? Go back and look at that. Know the microscope parts and their functions. Understand the difference between total magnification and just magnification. Know what resolution is. If the 10x objective lens is in place, what's the total magnification? So what is the magnification and what's the total magnification? What's the function of the condenser? What's the function of the iris, diaphragm, lever? What magnification and total magnification would oil immersion be used with? And this is it for this lecture. So just go over those and make sure that you're really getting the difference between magnification and total magnification, and know that with 1000x total magnification, we always use oil.